Hey everyone, this is Mrs. Miller. I'm going to talk to you today about enzymes and activation energy. So, yesterday you saw a couple of demonstrations about the amount of energy needed to start a chemical reaction. With the marshmallows, you saw that it took quite a bit of energy to make the chemical change happen. And then we talked about how it doesn't take that much energy to break down this marshmallow if you eat it. Then you saw an example of how a chemical reaction can be sped up using what's called a catalyst. So before we go on, I want to remind you that enzymes are proteins, and so they're going to have those characteristics that you learned a few weeks ago when we talked about the different organic compounds. So energy is the power required to change or move matter. There are lots of different types of energy. There is chemical energy, mechanical energy, thermal energy, nuclear energy, but what's the ultimate source of all energy? Where does all energy come from? From the sun. Now, living things store energy in a molecule called ATP. When you eat food, your body converts those organic compounds into ATP. And then when your cells need to do work, they can break that molecule down in order to release the energy that's inside. So think of ATP as sort of the energy currency of the cell. You need a lot of it to get anything done. So in a chemical reaction, we have two parts. What goes into the equation is called the reactants. What comes out of the equation is called the products. And the sum of all the chemical reactions in your body is called metabolism. So what this means is that when you eat food, all those chemical reactions that are taking place break down the food into their simplest molecules and then rebuild them into the parts that make you. So literally, you are what you eat. You break down the foods that you consume to convert it into your cell parts. So what do you need to start a chemical reaction? We talked a little bit about this yesterday in our demonstrations. The energy needed to start a reaction is called activation energy. And look here, notice that there is a lot of energy required before this reaction can release its products. I like to think of the analogy of a soda machine. When you want to get a Coke out of the machine, you have to put in a full $1.25 or $1.50, whatever soda machines cost these days. You can't put in 75 cents and just get half a Coke, right? It doesn't work that way. Well, chemical reactions are the same. You can't put in half of the energy and get half of a reaction. If you don't have enough energy, the reaction doesn't take place. Well, so how do we carry out all of the chemical reactions that we need in order to function? We need a catalyst. A catalyst lowers the activation energy. That means it takes less energy to complete the chemical reactions that go on in our body, which means they can go faster. The example that you saw yesterday was of the hydrogen peroxide. Now, if I leave that bottle of peroxide open, it'll break down into water and oxygen all on its own. But that's a slow process. So I added the catalyst, potassium iodide, and that allowed the reaction to take place even quicker. So here we have two reactions. The one in yellow is a reaction with no catalyst present. Notice how much energy is required before it takes place. The purple is a reaction with a catalyst. It doesn't require as much energy. But notice that they got the same product. So if I went back to my soda analogy, a catalyst is almost like somebody hacks into the soda machine and makes it so that half the money gets you a whole Coke. An enzyme is an organic catalyst. That means they're found in living things. So this catalyst that I used yesterday, potassium iodide, is not found in living things. Enzymes are the same as catalysts. So how do enzymes work? Well, The analogy of a lock and key actually works two ways. First of all, when you put your key in your front door and turn it, what happens? The door opens. But if you put your key in your neighbor's front door and turn it, what's going to happen? Hopefully nothing, right? So an enzyme works like a lock and a key because an enzyme fits to a molecule called a substrate. And when the enzyme and substrate come together, just like a lock and key, a reaction happens. The other way that enzymes and substrates are like locks and keys is that they're specific. Just like there's only one key for your front door, there is only one substrate for each enzyme. So here is an animation of an enzyme and a substrate fitting together. So what you see here is that the substrate, the little blue triangle, fits into the enzyme in what's called its active site. Notice that the enzyme changes its shape slightly to fit with the substrate. And then what happens? We have a chemical reaction. The substrate is broken apart. Now, this can happen all kinds of ways. You could have two substrates that are put together. But in any case, the enzyme and the substrate work together to make the chemical reaction happen. Now, enzymes can be affected by lots of different factors. They can be affected by the temperature. If an enzyme gets too hot, it can go through changes called denaturation. That means they lose their shape. And if they lose their shape, it's kind of like if you get a puzzle piece wet. Have you ever had a cardboard puzzle and just one piece of it gets wet? It won't fit in the puzzle anymore, right? If an enzyme gets too hot, it loses its shape. And if it loses its shape, it can't do its job anymore. Enzymes can also be slowed down if their temperature is too low. Enzymes are also affected by pH. Now, pH is a measure of how acidic or basic something is. Very strong acids are here on the left side of the screen, and they include things like battery acid, but a weak acid might be something like lemon juice. Neutral pH is right in the middle at 7. Your blood has a pH of about 7.5. So most of the enzymes that are in you function at about this level. And then very strong bases include things like bleach, but weak bases might include things like milk. And then finally, enzymes are affected by their relative concentration. And all that means is how many active sites are present on the enzyme. The more enzymes there are, the more reactions can take place. That's all I have for now. I hope that this was helpful, and I will see you tomorrow.